Very good morning to all of you. Department of Chemistry started this function yesterday, and we fruitfully learned something about nanomaterials, their types, some applications. Today is the second day, and now I invite Professor Raj to give his talk on nanomaterials, pollutants and remediations. So I hope you will enjoy this talk and learn accordingly. Thank you.
Wait. Yeah. Ooh. Okay. Okay. Good morning, everybody. Today we will continue our journey in nanotechnology and environmental applications of nanotechnology. We'll talk about different subjects today. There's also a tutorial in the afternoon. I'm still not sure what we will do in the tutorial. I will think maybe we will do something interesting. Okay, so the first lecture will be about good things and bad things. We'll start with the bad things. We will talk about air pollution from nanoparticles. In particular, we'll talk about toxicity of nanoparticles, why they can be bad for our health. In particular, we'll talk about a very big environmental problem, which is plastic. There's plastic everywhere, but there's really bad problems with small particles of plastics, particularly microplastics and nanoplastics, and we will discuss this. But then we'll talk about the good things, how nanomaterials can help us, how it can help us, for example, or specifically, to clean water, which is something very important. Okay, nanoparticles and why they are bad. If you think about it, we are exposed all the time to nanomaterials. In addition to nanomaterials, we are exposed also to other pollutants, like bad things that's in the air. There is exhaust from cars. There is things that get released when there is wind. There are things that are in the water that are very small and can cause health. Sometimes it can be even worse when you have nanoparticles in the air or in the water which combine with other pollutants, molecules, toxic molecules that's in the air or in the water. And when they are combined together, that can be even worse. Let's see why it is worse. Let's first talk about the air. You know, if you take very small particles, they can float in the air just like dust. And they can go anywhere, including into our body, through our lungs. Do you know what it is? It's called PM2.5. PM2.5 particle matter or particulate matter 2.5. These are particles that have diameter of approximately 2.5 microns. Everybody talk about PM2.5 because they go into our lungs And then from the lungs, they can spread throughout our body and cause damage. So actually, when you see numbers or parameters for air pollution, people usually talk about PM 2.5. There are apps on the phone that can tell you any minute what is the concentration of PM 2.5 in the air. It can give us an idea if it's safe to be outside or not safe to be outside. But if you think about it, look, PM 2.5 compared to nanoparticles is huge. It's like a huge ball compared to a small ball. So if this is a problem, because this goes into our body and causes problems, then these small nanoparticles are even worse. Because it's very easy for them to go into our lungs and then go into our blood, go into our brains, go into our different organs. That's the big danger about nanoparticles in the air. Actually, nanoparticles in the air can come from car exhaust. They can come from industrial plants. They can come from waste. They can just come from dust if you step on the ground. Actually, in the modern world, there's other sources of nanoparticles in the air. For example, 3D printers. You know, 3D printers are very big industry right now. Sometimes people take 3D printers and put them in their homes. But when you print something using 3D printer, the polymers, plastics that you use for printing can go in the air, fill the room, and this is pollution. This is air pollution. Metallic nanoparticles. These are metallic nanoparticles. They actually be surprising that we can have metallic particles in the air, like aluminum, like titanium, iron, silicon. But all such particles, metallic particles, can come from various sources. For example, from industrial plants. A plant that makes cars, make engine, anything that you can use metal, iron, steel, all these plants can release metallic nanoparticles in the air. And then these metallic nanoparticles can go and penetrate into the cells of our body. What I'm going to show you in the next few slides is that why these metallic nanoparticles are so bad for you. So let's see the first reason. The first reason why metallic nanoparticles are bad for you is because, first, because they are so small. MNP stands for metallic nanoparticles. Because metallic nanoparticles are so small, they can penetrate very easily through the membrane of the cell. Now, when these metallic nanoparticles enter the cell, it's very bad. In our cells, we don't have metals. We don't have metallic nanoparticles. When they go in, they can do bad things. They can go to different organelles in the cell. There are special compartments in cells that are very important for our biology. For example, the lysosome, the mitochondria. Metallic nanoparticles can go inside these organelles, and then they disrupt their normal function. And then we can have disease. Another thing they can do which is actually one of the main problems with metallic nanoparticles. They can oxidize. And when you have oxidation of biological molecules, you can have what we call ROS. When you see ROS, which stands for reactive oxidative species, it's very bad, because ROS can really cause damage in biological processes and can cause various diseases, primarily cancer. So these are nanoparticles in the air. We're going to talk about these ROS and oxidation in a minute. But first, another general survey. Nanoparticles can go to cells. If we look in the bigger picture, nanoparticles can go through the lungs to our brain. And when we have nanoparticles in our brain, it's very bad. It hurts our thinking. It hurts our cognition. Now, how nanoparticles can go to the blood, to the brain? Nanoparticles can go to the brain because they're so small, they can penetrate this thing which is called the blood-brain barrier. Have you heard of the blood-brain barrier? The blood-brain barrier, which sometimes people call the BBB, okay? This is an extremely important membrane that protects the brain from the rest of our body. We don't want anything to go into the brain because it's the most important organ in our body. So, to protect the brain, we have a very strong and tight membrane between the brain and the blood vessels. And this membrane, this layer, prevents anything from going into the brain. Only very small molecules. But because nanoparticles are so small, they can penetrate through the blood-brain barrier and enter the brain. And then this is very bad for us. Another particle or another type of materials that can be very bad for our health is microplastics and nanoplastics. You know, we are all surrounded by plastic. It's everywhere. From our water bottles, we don't have water bottles here, water bottles, our chairs, our shirts, our textile. Everything is plastic. And when we take plastics and we wait 1 month, we wait 1 year, we wait 10 years. Here, we have plastics. Very important because it has water. Thank you. So what happened to the plastic over time? It breaks. And it breaks. And it breaks some more. And eventually, what we have are tiny particles of plastics that are everywhere. People found plastics, for example, in tissues. This is a microscopy image, okay, of a tissue from some animal. And you see, this is the tissue. These things are microplastic particles. So think about it, some tissue in our brain, our heart, our brain, whatever, with small plastics. It's very bad. So this is where the micro and nanoplastics comes from. The bottle is breaking and releasing these very small nanoplastics. And these plastics, these small plastic particles, can actually break even more into molecules, which are very bad because they come from oil, from gas. Then they go, for example, to fish. Now, these days, when we take fish from the ocean, even far away from civilization, in the Pacific or in the Indian Ocean, we found that fish has plastics, microplastics in their bodies. This is, for example, shellfish. Okay, something that we can find on the shore. This is a shellfish, and when scientists look very close on the surface of the shellfish, they found these tiny plastic particles. So think about it. Microplastic particles, nanoplastics are everywhere. And when they go into our body, again, they can really disrupt and break important biological processes. This is what polystyrene particles, nanoplastics or nanoparticles, do when they go inside neurons in our brain. You know, polystyrene is maybe the most common plastics. Polystyrene, you can find it in sheets of nylon, sheets of plastics that you use for wrapping things, in packages. Now, what scientists found is that nanoplastics of polystyrene can enter into various neurons, and then what they do two things. One thing is when these nanoplastics goes into the neurons, they kill the neurons. The neurons die. If the neurons die, it's not good for us. It affects our thinking. It can affect dementia. It can make us sick. Another thing that they found about nanoplastics in neurons is that the neurons do not develop so well. And you know when it's very bad that neurons cannot develop well? In babies. When babies grow and become children, the neurons in the brain need to develop. That's very important for children to be smart. So scientists found that when these nanoplastics go into neurons, the neurons do not mature, do not develop well, affecting cognition of children. Do you know autism? Autistic children. Have you heard the term autism? This is a problem of communication. You know that in the last 30 years, the abundance of autism increased almost 30 times. There is many, many more autistic children in the world, at least in Western countries. And people don't know why. People don't know why there's so many autistic children. Maybe this is the reason. Because there's so many nanoplastics, so much nanoplastic pollution that children brains do not develop well and they become autistic. So this is the bad things about about nanoparticles in general. Now what about the good things? I told you that this course will present bad things about nanotechnology in the environment, but also good things about nanotechnology in the environment. And now, after we saw the bad things, let's have some hope. Let's see the good things. So the good things about nanotechnology in the environment is that nanomaterials can be used for cleaning the environment, specifically cleaning pollution from other nanomaterials. What we see here in this slide is a general description of the good things that nanomaterials and nanotechnology can do to the environment. One thing, absorption. As we're going to see in a minute, nanomaterials can absorb bad molecules from the air or from the water. When they absorb them, they remove them from the environment. Filtration. Nanomaterials can use as filters, very small filters, for bad nanomaterials in water. Oxidation. We're going to see about oxidation, but oxidation can remove also and break bad molecules in water. Then there is also photocatalysis and disinfection. We're going to talk a little bit about each of these good things that nanotechnology can do for us. Let's start with absorption. You know, in the water, there can be many pollutants, organic pollutants. Organic pollutants can come from the soil, for example, okay? When you use materials for enhancing growth of plants, of crops, or when you have cows and other animals on the ground that can pollute the ground and pollute the water. You can have pollution from industry. You can have pollution from people. So the risk of water pollution is very significant. What nanoparticles can do for us? Nanoparticles are very small, so they have a big surface area, relatively speaking. So we can take nanoparticles, and they can, just like a sponge, when you wash yourself in the shower, nanoparticles can absorb these pollutants from the water, and then when they absorb, you can take them out of the water, for example, by precipitating them from the water, and then you have clean water because all the bad molecules absorbed to the nanoparticles and were removed from the water. This is just a nice visual example of these nano cleansers. These nanoparticles here absorbing bad molecules on their surface and then removing them from the water. These are examples of some actual nanoabsorbers that people use. Some of them are even commercial products. So we have here polymeric sheets. These are nano sheets from polymers. We can have carbon nanotubes, zeolites that are special materials with lots of surface area, and metal nanoadsorbers. All these nanomaterials can just absorb pollutants and remove them from water. Nanofiltration. Nanofiltration is another very useful application of nanomaterials for cleaning water. This is just like a filter. For example, when you want to use a filter in the kitchen, you take whatever, water, and you pass it through a filter, and something cleaner comes out on the other side. This is exactly what nanofilters are doing, but they're doing it on the nanoscale, very small. So what we see here, these are materials that cause very small holes, and through this nanofilter, you can absorb all the bad things in the water, and what comes out on the other side, clean water. Nanofiltration. What you can remove from the water, you can remove molecules, you can remove bacteria, you can remove nanoparticles. Everything that you don't want, you can filter through this nanofiltration. Oxidation. In many cases, you can take nanomaterials which will oxidize pollutants. Now, what is oxidation? For those of you who are not familiar with oxidation reduction, these are very important chemical reactions in which electrons go from one molecule or from one species to another species. So, for example, we have an atom here. When it loses an electron, we say that this atom was oxidized, this atom was reduced. Now, these oxidation reduction reactions are very important for many chemical processes. For us, in this course, oxidation is very important because it can break organic molecules and organic pollutants. This is an example. You see, this is a molecule called magnesium dioxide. It's a very good oxidant. It takes electrons. So when we mix magnesium dioxide with, for example, oxygen, and then there is an organic pollutant, what this molecule is doing, it oxidizes this pollutant, and through this oxidation, it breaks many chemical bonds. So you see, you take this pollutant, very bad, and you break it through oxidation to these small molecules that are okay. Sometimes they're even attractive molecules, like NO3 minus. Nitrate is a very important molecule for enrichment of the soil. So what I want to tell you here is that oxidation is a very important chemical process for breaking toxic organic molecules in the water. Another version of this oxidation reaction is through photocatalysis. We're going to talk about photocatalysis a couple other times in this course. When you have photocatalysis, in many cases, you have this reduction oxidation reaction induced by light. Light is shining on a molecule or atom or a nanoparticle. What the light is doing, light is giving energy to this particle, to the molecule, and the molecule can take this energy and does oxidation. So we see the oxidation here, and again, when you have oxidation, you can break organic pollutants in the water. So what did we learn? We learned that nanoparticles can be a health hazard because of their size. We learn that nanoparticles can accumulate in different organs in our body, in the lung, in the brain. We talked about why metal nanoparticles are bad for us, specifically because of the oxidation reactions of metals. But we also talked about nanomaterials can help us to remediate pollution. How we do that, how nanomaterials do that, they can use as nanoadsorbents. They can absorb molecules and pollutants in water. They can use as filter. We can have nanofiltration through cleaning the water through absorbing bad species, just like any other filters. They can use for pollutant oxidation. We can oxidize the pollutant through redox reaction, through reduction oxidation reaction that can happen in metal nanoparticles or through photocatalytic reactions, taking the light and doing reduction and oxidation. Questions? Okay. So we will take, yeah, sorry, yeah.
Sir, we did great lecture. You have really covered. And it's a common problem. Common people,
Like?
microplastics.
That's right.
So my question is that, so what's the process to have this microplastics? So because of the degradation behavior of the plastics, how we get that?
Okay. Microplastics are what we get when plastic over time breaks. It degrades because the bonds in the plastics, in the polymers, are not very, very strong. So over time, sometimes it takes days, sometimes it takes years, the plastics just breaks. And then from big plastics, you have small plastics. And from small pieces of plastics, you will get small microplastics. And then from a small microplastics, you can get nanoplastics.
So if we suppose, if we get plastic which is not degradable at all,
Okay.
polyethylene, so is there a chances to get microplastics?
To get less microplastic. You mean if it's non-degradable?
Not degradable at all.
That's actually a good point. Because what we want, we're always talking about biodegradable plastics, which are very good. But you're right that if you don't take biodegradable, you will get less breakage of the plastics. But the generation of microplastics is mostly through mechanical forces, through breaking by pulling, by the wind, by the water. So even if you take non-biodegradable plastic, still you will get microplastics and nanoplastics just from mechanical forces, like breaking with your hands or by cars or whatever.
Like this plastic.
Yeah.
Water is made up of PET.
PET, poly, yeah.
And the ester bond.
Yes,
Because of the ester bond, that's the reason it is degraded.
that's right.
So can we make such model of polyethylene to save the environment?
To what?
To save the environment?
Well, maybe it will be better, but any plastics eventually will break into small pieces mechanically. And when you have small pieces, like you get microplastics and nanoplastics, and they can go into your body and they can cause damage. So, yeah.
Yeah, your lecture indeed is excellent. I like the notes that you have covered. It's not only for the chemist, but also for the common people.
Yeah. So it's good or bad?
How they because everything, what we are getting from the market, household items, we are getting, we are unaware
Yes.
how much microplastic we have.
Absolutely.
So how to educate common people regarding this problem?
Okay. Very good question. How can we make changes in our life to help us with microplastics? So one thing you can do, first of all, try to minimize consumption of plastics. That's the one thing that's important. Let's not make so many bottles. The other thing is use, for example, for water, in Europe, for example, they use a metal bottles, multi-use containers for water. So then if you finish this bottle, you don't go and buy a new bottle, which might be bad, but you fill a metal container and then you use it 100 times or 1,000 times. That's what you can do, for example. Another thing is indeed use biodegradable plastics. And biodegradable plastic, at least you can. The big problem, for example, with this is that when we finish using it and we throw it, it goes to a waste dump, or you find it on the side of the road, and then it's breaking slowly, and you get microplastics. So at least you're right. If you take biodegradable or degradable polymers or degradable plastics, then at least you don't get these accumulation of plastic waste and plastic trash.
It's very interesting. We can keep on discussing on these topics. The last question I take.
Sure, take your time.
Just how to detect microplastics? Is it any easy device?
The question was, for those of you who didn't hear, it's a very good question. How do you detect microplastics? Actually now, there's no easy way to detect microplastic. You can detect microplastics maybe in your lab, but it's not so useful. What I will describe in, I think, lecture number 8 or 9, when I talk about nano sensors for the environment, I will talk about the technology that we developed in our lab for detection of microplastics and nanoplastics in the air. So wait, be patient until Thursday, and we're going to talk about, I'm going to tell you about very interesting technology for detecting nanoplastics in the air. Yes?
We focus, let's say, on the microplastic, especially in the aqua compartment.
Yes.
Then, as the consequence of renewable energy sources in terms of, let's say, photocatalysis, the moment it can be commercialized or making it in a big level, then what do you comment, like whether the microplastic is going to be more concentrated or particular?
The question was about, I'm repeating your question because you're sitting in the front, maybe they don't hear. If there will be more microplastics when you have renewable energy application. I hope this was the question.
So it will be more concentrated as the consequences of one hand having the renewable energy and the other hand
Yes.
more concentrated.
Okay. So I'm actually not sure if there will be more renewable energy devices. I'm not sure if there will be more plastics in the water.
No, my point is that just consider like the device is making catalysis,
Yeah.
water is splitting, then it gets heat concentrated, and at the end you might be having maybe less amount,
Yes.
more concentrated
In small places. That's certainly possible. That's why if you study chemistry, you're going to have good jobs. Because there should be ways, we should develop new ways to solve this problem. You're right, there will be maybe good things about energy, but bad things about pollution of microplastics more concentrated in the area of the energy applications. We will have to find a solution. Sure.
So if we compare between the microplastic and PFAS material, because PFAS material is one of the unavoidable things which is used for the device materials rather than microplastic.
Sure.
So how do you take both in two hands and having the
Okay. So the question was about microplastics and PFAS. PFAS are what people called forever chemicals. These are molecules of polymer that do not break. They can stay in the water and in the soil forever. That's why they're very, very dangerous. Now, there's actually not a connection between the two things because PFAS are molecules and microplastics and nanoplastics are like aggregates of lots of plastics together. In both cases, we need to find ways to break these molecules, and that's how you can clean the water. But with PFAS, still it's very difficult to break the bonds. These are very stable molecules, and that's the problem about them. Because they're so stable, they don't break. So that's why you cannot recycle them. So there's many problems. There's many environmental problems. Our job, your job, is to find solution. More questions?
Any questions from students side? Yeah, please. Go ahead.
student take question? Question come on.
Yeah.
In recent years, there was a material that is hydrolysis sequences, which eat plastics, PT. Hydrolysis sequences which eat plastic. So, is there any chance that the microplastic can be eaten that we can develop in?
This is a very good point. There are actually now bacteria, for example, that can eat plastic, that they can hydrolyze PET in these bottles. Still, it's not so easy. You know, it takes time because there's bacteria, they cannot just sit here and eat the plastic. It will take them 1,000 years. But there are people that are working on development of special bacteria. Yeah. I'm sorry? Of course. It's very expensive. And bacteria are very... Yeah, you're right. But think of... Yeah.
So we can culture in the lab also. Because then in the
That's wonderful. But think about it. You have every day millions of bottles everywhere. So it's not so efficient. If you culture some bacteria, to get rid of all these bottles, you need millions of agar plates. So this, what you do is very true. You can really culture bacteria. You can grow bacteria that eat plastic. But the secret will be to make it commercial, to make it cheap, to make it very useful. This is the challenge.
From the bacteria, there are some reports of insects
Oh, really?
So not saying,
Interesting.
yeah, that's only that part. Imagination is more than reality.
Well, everything starts from imagination.
Yeah.
The good ideas starts from imagination.
Lots of work is going on embedded enzymes.
Exactly. Actually, the bacteria that break the PET, that hydrolyze the PET, they use enzymes. And some people, they take enzymes and they use them for breaking plastic. But enzymes are even more problematic because enzymes are biological molecules. They're very sensitive and they're even more expensive. But there are many ideas, as you see. We need to get rid of the microplastics and the nanoplastics. And there's many ways to do that. Okay, so we will take 10 minutes break.
Yes, sir. Yes.
Let's take 10-minute break and continue for the next lecture.
Well, on behalf of Department of Chemistry, I thank you, Mr. Raj, for your first lecture today.
Third lecture.
First lecture today.
Ah, the first lecture for
That's
today.
what I'm saying.
Yeah, I thought you said the
Please,
fourth,
please
sorry.
take your seat.
Yeah.
And I hope our students have benefited from this lecture. And we had some queries, and there was a and I wish the students actually for whom nanomaterials. So, please.
Yeah. Okay. So, the previous lecture was bad things and good things. Remember? This lecture is only going to be about good things. It's always better to talk about good things and not bad things. So, the good things we will talk today, now, is about how nanomaterials can help agriculture. Mostly agriculture, what people sometimes call green nanomaterials. Green is something very attractive and very good. So what we will talk about is particularly nanomaterials against bacteria, against microbial infections. We're going to talk about agricultural applications. So the topics that we will cover in this lecture, we'll talk about green synthesis of nanoparticles, particularly metal nanoparticles. As we saw in the previous lecture, nanoparticles and metal nanoparticles are very useful. Many people use them, so we will discuss good way, green ways to make nanoparticles. Then we will talk about nanoparticles in agriculture. How nanomaterials and nanoparticles can help us to get better yields, to get better agriculture, and to get better life. We will particularly talk about very interesting and important material called nanocellulose. We're going to see why nanocellulose is so important, and we're going to see where we can use nanocellulose and in what applications. And then at the end, we'll talk a little bit about carbon nanoparticles, which are very useful. And we're going to meet them throughout the course in other lectures also. Okay, let's start with making nanoparticles from agricultural sources, from green sources. So green sources is a general term for everything that is natural. Things that you can find in the fields, things that you can find when you grow something, things that you don't need to do chemistry to produce. This is the general title, green materials. So we're going to talk about green sources and all the good things that green sources contain. And we will describe some ways of how you take all these good things from green sources and make nanoparticles. And remember, if you make nanoparticles, you can do important things, good things with the nanoparticles. So this step is very important. And that's what we're going to talk about in the next few minutes. Let's talk specifically about metal nanoparticles. You remember from the previous lectures, we talked about metal nanoparticles. These are the particles that you can use for example, oxidation, cleaning water from pollutants. So metal particles are very useful. And if you see all these things, you never thought that you can take honey, and from honey you can make metal nanoparticles. Or you can take fruit and vegetables, and from fruit and vegetables you can make metal nanoparticles. But this is true. What I'm going to show you is one or two processes that people can take these natural things and make metal nanoparticles. It's very interesting. As we saw before, metal nanoparticles can be very good for cleaning water, for example, doing oxidation and reduction. And just to remind you again, why metal nanoparticles are important. Metal nanoparticles can be used, for example, for redox reaction, reduction and oxidation. This slide is just to remind you why we want metal nanoparticles. Actually, in particular, it wasn't on. Now it is. Ooh. Did you hear all what I said? Okay. Now I can speak much softer. Just don't fall asleep. Okay, so let's see the process of making metal nanoparticles from plants. So all these things, these are sources of important molecules that can help us to make metal nanoparticles. So we put this in water, we can grind it, we can dissolve it. What's important is in this water, we're going to have all these important biological materials. Then we will put salt. Salt, as a general term, is just metal ions and their counter ions. For example, sodium plus, chlor minus, iron plus, chlor minus. All these are salts. Metal salts is something very cheap. You can buy iron chloride. You can buy magnesium acetate. You can buy salt very cheaply. So you put the salt in, and the amazing thing that can happen in this solution is that the biological molecules and enzymes from here, from the organic material, what they can do, they can reduce turns off all the time.
I think that button is getting switched off. So you better keep it. Do you have a pocket here?
No?
Look that there.
Maybe I'll just stand here like this.
You can't do that. You know, it's because it's your nature.
No. I'm always moving.
Yeah.
Okay. That's good. Thank you. Yes, I'll do that. Thanks. He's an engineer. Okay, so we make metal nanoparticles. This is another example how to make silver nanoparticles. Okay, again, this is a real experiment. These are real pictures that I took from some paper. If you don't, maybe you have something else. Every minute I'll press here. That's okay. Don't worry. Okay, so we take what it looks to me like a broccoli or something. We take these plant, okay, we grind it very well, and we make a powder. Okay, we take the Then you take the extract, the powder, and you mix it with salt. This is silver nitrate. It's a salt of silver. It's very cheap. You can buy it in the store. When you mix it together, and you get this red solution. And when you have red solution, you know that you have important ingredients that can help our health. For example, polyphenols. We have polyphenols, you know where? In green tea. Green tea has polyphenols. Polyphenols are known as antioxidants. They're very good for our health. Our health. Now, when we take metal nanoparticles in the presence of these molecules from plant extract, Maybe you don't know that, but cellulose is the most common carbon material on Earth, other than carbon dioxide, for example. All the plants in the world are made of cellulose. You have cellulose particularly in the bark, you have cellulose in the leaves, you have cellulose in every plant. You know why cellulose is so important for is because of this organization. Cellulose make very strong fibers, just like our shirts. Shirts also made of fibers. Plants are also made of cellulose fibers. And not only that, is that micro to nano, different landscape. This is structure, and this is what makes cellulose so useful for plants, and also so useful for us, because then we take the cellulose, we can make furniture, which are very strong, we can make paper, which is very thin. We can make filters. We can make clothes. We can make everything just from these cellulose nanofibers. Now, there are all these obvious applications of cellulose, but in the nano world, in nanotechnology, and here you can see some of the applications of nanocellulose. Remember, this lecture is about green nanomaterials. And nanocellulose is the creation. So there are many other things that you can make with cellulose nanofibers. This is just some examples that we can find, that I found one day for applications of find that you can use in buildings made of nanocellulose. These are coatings. Transparent film, you make a very, very thin layer of nanocellulose. If you make it so thin, light can go in cellulose. Now, I'll describe now some of the very interesting chemical assemblies for nano. Aerogels, we'll talk about them in a minute, and hydrogels, which are also very interesting assembly that we're going to talk in a minute. Let's talk about aerogel. Fantastic material that people make. The reason it's transparent and it's light is what you can see here. This is an electron microscopy image of aerogel made of nanocellulose fibers. See all these Now, this is a microscopy picture, but think about it. If you have this entire volume inside, it makes the material very flexible. You can stretch it, you can compress it, and it's not going to break. You can bend it, you can give it any shape you like. This is why hydrogels are very useful. The way hydrogels are built is basically you have a framework. In our case, the framework is made of nanocellulose fibers. And in the framework, you have all these water molecules. So this hydrogel has special properties. So this is just the skin, an example of nanocellulose hydrogel. So we have the nanofibers. Sometimes we have ions that we add. Okay? These are the building blocks, the cellulose building blocks. You have to add water. In this case, you start by ice crystals, and after the process, you end up with nanocellulose hydrogen. Look how cute it is. Just like something very flexible, press it, you do anything you want. You can have many types of applications with hydrogen. This is another example of useful carbon-based nanomaterial. This is called biochar. Biochar, usually, sometimes people look at it as waste, just carbon waste. Actually, biochar is also what you get after burning carbon materials. For example, burning trees, burning leaves. You have this powder, and the powder here contains many types of nanomaterials, nanoparticles. What you do with that, you get these nanoparticles. Here is biochar, which you can, actually it's called char. And when you absorb various biomolecules, you get nanoparticles, biochar. Useful material. So we talk about nanocellulose, we talk about char or biochar. Let's talk about very important class of carbon nanoparticles, which are actually very close to my heart, because I work with these carbon nanoparticles in my lab, and I'll show you this week some very interesting applications that we do with these carbon nanoparticles. Actually, these carbon nanoparticles, which are called carbon dots. Why I call them dots? Because they're very, very small. And the way to make carbon dots is actually extremely simple. You don't need to be a top scientist to make carbon dots. Basically, what you do, you can take these materials. You know what's common about these materials? Honey, banana, beans, candle, juice. What's common about all these? Carbon, exactly. All these materials contain carbon. If you take this material and you heat them, not such a high temperature, 100 degrees, you can do it at home in a stove.
Sir, can we have your mic?
What?
We can change it.
Maybe better. Okay, thank you. It will help me with my voice. Okay, I continue now and down. You take the carbon source and you heat it. Not high temperature. Maybe a few hours. When you look at what you get, you see that you have carbon dots. You look under the microscope, you see small nanoparticles of carbon, which are very interesting. This is actually the structure of these carbon dots. You have these carbon regions and different groups on the surface. This is the internal structure of the carbon dots. Thank you. Thanks. Let's hope it's not going to stop. So again, these are other sources. Oop, what happened here? Yeah. Okay. So this is how we make carbon dots. What you can do with carbon dots? You can take carbon dots, okay, and by absorbing light, the carbon dots can help photosynthesis, for example. How they help photosynthesis? Because carbon dots, they're so small, and they have these different groups on the surface. They can absorb light and release the light and electrons for the plants to help with the photosynthesis. So these are just examples of what you can do with carbon dots in agriculture, again, as a simple carbon source that plants can use both for food and for photosynthesis. So you shine light, there's electrons that get released from the carbon dots to the chloroplast, the molecules that help plants to do photosynthesis, and that's how the plants can use it and generate important biological molecules in order to grow. Carbon dots also can be used as delivery system, just like trucks, delivering important ions and nutrients to plants. So this is the framework of the carbon dot. And all these little things here are just ions that are important for plants, like sulfur, like phosphorus, like nitrogen, like potassium. All these molecules all these ions and atoms are important for plants. They can absorb into the carbon dots, and then plants use the carbon dots, simply eat the carbon dots for their uses. Actually, there were some scientists that showed that you can also take genetic materials, DNA. You can take small DNA, absorb the DNA inside the carbon nanoparticle, the carbon dot, and then deliver the DNA to plants in order to make the plants healthier. Again, the key thing is that because carbon dots are made of carbon, they are okay for the plants. They're not toxic. They're not like metal nanoparticles. They're just carbon, just like food. Food for us and food for the plants. So using the carbon dots, you can deliver to the plant nutrient, you can deliver genes to the plant, and you can deliver antibacterial and herbicide, all through the platform, the delivery platform of carbon dots. This is just an example of a research where the people showed that actually they took carbon dots, they mix it with chloroplast. Chloroplast are the organelles, the proteins in the plants that are responsible for photosynthesis, and they found that the carbon dots go in the chloroplast and help cells with photosynthesis. Okay? You can see here, these are just plants. With carbon dots, the more carbon dots that you put, you have better growth of the plant. So carbon dots just help plants with their photosynthesis. And that's the end. Actually, I missed something here, but that's the end of this story. Thank you. Yeah, there's one slide that I was missing here, but next time.
Second lecture has completed. If any one of you have any questions, please come up.
Oh, yeah. Sorry. Of what?
What are the application of metallogels?
Of metallogels. Okay. So I guess what you mean are gels with metals inside. Okay. So in some cases, gels in general, people like them because they have a lot of pores inside, a lot of space that you can put other molecules, guest molecules. Metallogels are special gels with a lot of metal ions and metals in general. In some cases, you can use metallogels to deliver metals to targets. For example, you need metal in certain plants. You might need metals in certain parts of your body where you need the metals. In some cases, you just want a gel that will be very stable. And metals, if they are in the scaffold, the framework of the gel, they can make the gel very stable. So this is a special subfamily of gels, metallogels. Yes?
You have shown that in that silver synthesis, red color solution is here, but the silver ion is colorless. So why that was red?
Okay. Why it was red? Let's go to this slide and explain why it was red. I just repeat the question for everybody. 1 minute. Let's see. It's this slide here. So, yeah, the question that he was asking, silver is colorless. The extract from the plant is green. How come this is red? This is actually the special property of silver nanoparticles. Silver nanoparticles, they absorb light and they become red. It's actually called plasmonic effect. And the red color actually tells us that in the solution we have not silver ions, but silver nanoparticles. This is actually something that you can do with silver nanoparticles. You can make colors for certain uses. For example, photography. You know, in photography, in old-time photography, what people do, you had a film with silver ions. And then you add a material for processing. It's not now that you have picture instantly on the cell phone. 30 years ago, you went to a shop. You take picture with a camera, and you went to the shop to process the film and get pictures. And what you had on the film is silver ion. After you process, they became reduced, and you get silver nanoparticles, which has a color, a red color. Yes? Oh, sorry.
Regarding green color,
Yeah?
right?
Red color, yes.
Made with silver. But regarding
Not silver ion, but silver nanoparticles.
sorry.
Yeah.
But how about the other materials?
Yeah.
Because it's a mixture of polyphenols, terpenols, they may also impact.
That's certainly possible. It's if there are some other materials, they can oxidize or reduce or just absorb to the nanoparticles. Usually what people do after they make this reaction, they purify. They take the silver nanoparticles, they precipitate them, and they throw away everything else. But if you want polyphenols and all the other things, you can mix it together with the silver nanoparticles and then use it if you want. In this particular slide, all what I showed is a way to make from silver ion and extract, plant extract, how to make silver nanoparticle. Very simple process, green process. You didn't have to do anything else, just mixing. If what? You're right, it depends on the plant extract. Silver nanoparticles in general are red. Sometimes they change the color depending on the size. Big particles are more blue. Small particles are more red. But absolutely, depending on the extract, depending on the material, the molecules in the extract, you can get different colors. Yeah.
Actually, I teach this topic, so I would like to add one thing. When the bulk material goes to the nanomaterial, the optical as well as the electrical property change. What sir is saying, the size dependent change in color. Okay? So that is true. When it goes in the nano level, the silver ion, which is colorless in the ion form, but when it forms the nanomaterial, which may have 100 atoms, which may have 1000 atoms, there are several models, the optical property changes. You understand?
So that is detailed.
And number one. Number two, that a coating. Coating is also what we say protecting agent or capping agent. That is equally affects the color of the nanomaterial as well as pH is also there. So, and in semiconducting device, this also we read in electrical property. So there is the size dependent change in its ACR. If you write in Google ACR, surface enhanced resonance, and based on surface enhanced resonance, there are tremendous application in sensing device. In MSc first sem we have nanochemistry and nanomaterial. So just write ACR, surface enhanced resonance. There you will find the absorption spectroscopy, UV visual absorption spectroscopy, as well as the fluorescence spectroscopy. What it appears, that is the fluorescence emission spectra, and what it absorbs, that is the absorption spectra. So what it appears here, red color. So that has different wavelength. What is being absorbed, that has different wavelength. Okay? So that's
Yeah. No, that's great. Actually, in a lecture in tomorrow or on Thursday, we will discuss the size dependence colors. I will mention that. Yeah. Carbon dots change color for one reason. We will discuss it. Don't worry. Yes, sorry.
Thank you for the lecture. Sir, I have a doubt that in these synthesis of nanoparticles, metal nanoparticles, you said that we'll take any plant, we'll bring it, and then we'll add the salt to it. So from salt, we'll get the metal ions. And then
Metal, yeah, from one.
And then there's the aggregation which will be formed. So my question is that aggregation which is formed and is of metal ions. So that metal ions coming from the salt only or the plant only, or they will combine?
Okay, the story here is that the aggregates actually, let's find this relevant slide. The aggregates that I was talking, this one, yes? Yes, okay. Yeah, this is the slide. No, previous one. Let's see. This is good. Okay, that's even a better slide. The aggregates that I'm describing here, these are metal nanoparticles. Because what happened is, zero oxidation state. This is the extract that have all the molecules and enzymes that can reduce the salt. The salt is ions. In order to get these aggregates, you need the metals not to be ions, but to be zero oxidation state, to be elemental. So you need to give them electrons. The electrons come from here, from the extract. So you have ions, the ions get electrons, then they aggregate, and these aggregates are the metal nanoparticles. There were more questions. Yes.
So as we discussed the use of carbon plots in agriculture. So as we know, there are pros and cons of everything. So what are the possible environmental risks in agriculture due to carbon plots?
Okay. Carbon dots actually by themselves are not dangerous in agriculture. That's actually one of the good things about carbon dots, because these are just carbon. The problem is when carbon dots, there's two problems. One problem is that the carbon dots sometimes absorb other things from the environment that can be toxic for the plants. Because remember, carbon dots have high surface area, okay? We saw it in this picture here. Just this, okay? We saw this picture here of the carbon dots with all these things here. So sometimes carbon dots can absorb bad things if you're not careful. And then these bad things will go to the plants and then go to our body when we eat the plants. The other possibly dangerous thing is that scientists, they still don't know for sure if carbon dots are good or bad for us in our body. We think that they're not so bad. We are sure that they're not so bad as metal nanoparticles, for example. But still, there are risks, because these are small spheres, small nanoparticles. Maybe even though they are made of carbons, maybe they go in our body and they cause damage. So when you give carbon dots to plants, and after we eat the plants, we eat the carbon dots, maybe there are risks. But let me tell you something. When you eat meat, people found that when you eat cooked meat, anything cooked, if you drink beer, for example, if you eat bread, people found in all these things you have carbon dots. That's really one of the amazing things in the carbon dot world. People found carbon dots only about 30 years ago. 30 years ago, they discovered carbon dots in carbon materials. So there was a big thing, new carbon material. Then they found that you have carbon dots everywhere. So we eat meat, cooked meat, of course, hopefully. We eat bread, we drink beer. We have all these things, and thank God we're okay. We're not dead yet. So that means probably that carbon dots are okay. But of course, we need to do research and to find out whether carbon dots are good or bad. Yes?
My question is that in carbon quantum dots, when we most of the carbon quantum dots are amorphous in nature, or if yes, then how can we just change this in nature when we drive? When we try to drive, it seems amorphous. And if it is possible that we can have dry sample. Solid dry sample.
Okay, actually, the question is about carbon dots which are amorphous, and if you dry them, it's still amorphous. Well, there's big discussion what is really the structure of carbon dots. We're going to talk about carbon dot more in other lectures in this course. Now, of course, when you dry it, it becomes some amorphous material, just like general carbon thing. And then it may have different properties as the small carbon dots. It's a big, big field. Big.
Is it possible that we can synthesize the specific non-amorphous nature?
It is possible. Many people do that. The question is what you will do with amorphous versus crystalline carbon dots. And this is something that is possible. You not just need to do a lot of research.
So that will be due to some structure like graphite and diamond.
Yeah, it is. Carbon dots is a relative of diamond. That's what's amazing about carbon in general. It can form different morphologies, different structures. Diamond is one, carbon dot is another. I don't know if you remember in my first lecture I talked about fullerenes, these C60s, which is another carbon material. Carbon is amazing. Yeah.
The powder form, we can take some green sources. So it's possible to take all type of green substance, or it's just a condition.
Okay. Now, when you grind, you make powder. The question is, if we can take any green material, green source, and grind it and get good things, that was the question? Now, of course, you can take any plant and you make a powder and then you dissolve it and get different things. The question is, what are the things? You know, some plants are poison. So if you grind it, you don't want to deal with it. You don't want to drink it, you don't want to eat it. Some other plants are more good. They have all these polyphenols and all these good molecules that are important for health. So my answer is that it depends on your source, on your plant. From any plant you can get molecules and you can get materials and you can make nanoparticles. The question is, what are the plants, if they're good or bad? Yes?
In fact, it's where there is that concept of net zero, means carbon emission, minimize the carbon emission. So, there is a mixing of carbon. So, it is possible to mix carbon dots instead of carbon, and then reduce the carbon emissions, CO2 emissions.
Okay, the question is, if I understood the question correctly, about net emission. You know, it's very important to prevent carbon dioxide go to the atmosphere. We will discuss it a little bit in the future, so in the next lectures. So the question is if we can use carbon dots to reduce emission of CO2, for example, in the atmosphere. The answer is yes. Of course, carbon dots are very useful. So in principle, you can take any CO2 or carbon, other carbon, and instead of burning it or releasing it to the atmosphere, you can make carbon dots. But the problem is, every day we release to the atmosphere millions of tons of carbon. We don't need yet 1,000,000 tons of carbon dots. There's nothing you can do with this. okay? So, of course, if we can do useful things with carbon dioxide instead of burning it and throwing it to the atmosphere, it will be good. The question is, how? And of course, carbon dots are one example of something very useful that you can do with carbon. So that's why people like it, including myself. More questions? Okay? Oh, sure. Yes.
That talk was regarding cellulose.
Cellulose, yes.
Nanoparticle. How easy to deal with cellulose?
The question is how easy it is to work with nanocellulose or with cellulose in general. So it's not so easy like carbon dots. Carbon dots you can make in your kitchen. Nanocellulose is little more complicated, but it is possible. Basically, what you need to do, you need to take these plants and you need to break them and separate the nanocellulose. So in scientific papers, there are many protocols, many ways that you can isolate and use nanocellulose. So it's not kitchen work, but it's not a complicated work. It's something that you can do. Many people are working with nanocellulose. It's not so soluble, actually. This is one of the problems with nanocellulose. It's actually an advantage also, because you can make coatings from nanocellulose that will be resistant to water, because it's not dissolved in water. So this is something, for example, you can make coating that cannot be dirty, because all the water will go away. So, like everything, there's good and bad. But it is a very interesting material, and they're very, yeah, and there's lots of cellulose, and there's many applications with nanocellulose.
Sir, in your support, I want to say.
Yeah.
When you go into the laboratory, take silver like this, and just use lemon, and just split it. You will get the silver nanoparticle. The citric acid, which is
That's right.
present in the lemon, that will work as a reducing agent as well as a cutting agent. Or else what chemists will do, we use sodium borohydride as a reducing agent. So here, I think as I understand, sir has taken all the time green sources, so that green sources used as a reducing agent as well as a cutting agent. Am I right?
Yes.
Yes.
It's a beautiful experiment. You can try.
You are chemistry student, so you should know the whole mechanism also.
So same thing will work with orange. Same thing will work with orange.
That I have not tried.
Less.
Citric acid should be there. Citric acid should be there.
There's more question. Yeah. Yeah.
Sir, as we know, heavy metals have very bad effects on the body, and it can last many years.
Yeah.
More than other nanoplastics. So my question is, potent metal nanoparticles. So can it say harmful for our body? Same like that, or and is there any degradation process or mechanism regarding this?
Okay. The question is about metal nanoparticles. Are they bad for us just like heavy metals? Now, of course, as I showed in the beginning, in I think the previous lecture, metal nanoparticles can go into our tissues and cells. They can reduce or oxidize, and then they can cause damage because they can generate oxidative species, reactive oxidative species, so they can be very bad for us. There are ways to break them and clean them which is also something that we can develop. They can be sometimes even more dangerous than the metal nanoparticles themselves.
Okay, I thank you, Dr. Raj, for your very inspiring lecture. And I think the students have, it was indeed a very inspiring and interesting lecture. It's a common, actually, topic. And a lot of things can be done out of these green materials. So, number one is what are green materials? Number two, how can you use it and where you can use it? So, it's a very, very big topic, very vast topic. So, I thank you, sir, for giving my students this knowledge. And now we'll take a break for lunch, and the next session will start at 2 p.m. Right?
आना है सबको, ऐसा नहीं लंच के बाद भाग गए.
आने के बाद तो सोएंगे।
Yeah, one more announcement that students, those have registered here. So make sure that you have signed. Okay? Infinity. Up, up, up.
Okay. So hello everybody. I hope you had a good lunch, good rest. This lecture will be about nanotechnology for water purification. After this lecture, we should have a tutorial, but I told Professor Patra that I don't have any tutorials, so we're going to have an open discussion. If you want to discuss something, I can tell you about other research projects in my lab, whatever you want to do. But I don't have a tutorial. Anyway, so the lecture of now will be about nanotechnology for water purification. In this lecture, we will talk about some things that I already discussed this morning. So some concepts you might be already familiar with. For example, carbon dots. We will talk a little bit more in details about carbon dots and how beautiful things we can do with carbon dots. But we will start with filters, nanofilters. Two-dimensional nanofilters will introduce interesting nanomaterials in two-dimensional films that we can use for water filtration. Then we'll talk about metal nanoparticles for water purification. And then I'll describe actually some things that we did in my lab in the last few years using carbon dots for water cleaning in general. Okay, how we clean water. So the easiest thing to clean water, if you think about it, is just like we do anyway. You have a filter, you take water from one side, the filter is supposed to collect all the dirt, all the pollution, all the bad things in the water, and then on the other side, you're supposed to have drinking water. Actually, these kind of filters are very common in many water cleaning devices and water cleaning instruments. Sometimes you can buy them in the shop. What nanotechnology can give us are special nano filters. And the nano here, the small dimension, what it gives us is a way to do excellent filtration, to filter the smallest particles, to be able to filter all the bad molecules. It's just very efficient filters. This is actually one of the most interesting two-dimensional nanofilters. This is called graphene. Graphene is also a form of carbon. Today we talked a lot about carbon. Why we like carbon? Because it's cheap, we can find it everywhere, and good for us. We can make very easily, or relatively easily, we can make different things from carbon. This is one of the things that we can make from carbon, graphene. Now, graphene is just a big sheet of carbon that connected in a hexagonal way in a 6-membered ring. And what's special about graphene, there's many things that are special, but the most important properties of graphene is that it's very, very thin. It's so thin that the thickness is sometimes, depending on the graphene, 1 or 2 or 3 atoms. So basically you can take many carbon atoms in the 2 dimensions. In the 3rd dimension, it's very, very thin. That actually makes graphene sheets very special, very light for one thing. You can easily bend them without breaking, just like you can see here. This is actually a real picture of a graphene sheet. Now, obviously, this graphene sheet is not 1 or 2 atoms thick. Otherwise, we will not be able to see that. But still, this graphene that this person is holding is less than 10 micron. Very, very thin. The other nice things about graphene is that it is extremely strong. Very difficult to break it and to tear it. This is because of the special chemical properties and chemical organization of the carbon. All these bonds, these double bonds, are very strong. So overall, the graphene is very, very strong and not breakable. The other thing that's remarkable about graphene, and actually we use for water purification, is that it is conductive. It's very conductive. It's conductive for electrons, so it can conduct electricity. In a minute, I'll show you why it is important. But actually, one of the applications that people use these days with graphene is just for electronics, carbon electronics, because it is such a good conductor. So how can we use graphene as a filter? Now, first of all, it's very thin, so we can use it as a membrane to filter. Now, because the openings here are very small, basically what we have is that water can go through these holes here. Okay, see a drop of water. But if you have bigger things, like nanoparticles, like microparticles, remember those nanoplastics and microplastics that we discussed before lunch? So when they go in, they can just get stuck here and don't go through. The same for other things, contaminants that might be in the water. Now, because graphene is so strong, so stable, you can pass water with pressure. It will do the filtration, but it will not break, which is something very important. Now, another very important thing about graphene as a filter is the fact that it is conductive. Remember, we said that graphene can conduct electricity. Now, why it is important? Remember that we talked before lunch about oxidation. When we have pollutants in the water, we can oxidize them, and when we oxidize them, we break them. Now, because graphene is such a good conductor, we can connect it like this to some kind of electrolyzer. We can pass electrons in these graphene sheet, and then when you have pollutant molecules coming closer to the graphene, they can get oxidized. They can get oxidized, and then we can remove them from solution. We break the pollutants, we degrade the pollutants, and remove them. So graphene can both filter and also use for oxidation of organic pollutants. Very effective filtration. Actually, the other nice thing about graphene, and graphene conductivity in particular, is that when we have ions, metal ions, somebody before lunch asked about heavy metal ions, like iron, copper, lead, all these really bad things that you can find in the water. Now, these ions can be in the water. Again, when they get closer to the graphene sheet, and graphene has all these electrons, you can get the electrons and reduce the positive ion or oxidize the negative ions. And then what happens is that these ions, because of the reduction or the oxidation, they become metallic and they aggregate and they sit on the graphene and they don't go through. So that's why graphene is such a good membrane, such a good filter. It can filter out particles, it can oxidize organic pollutants, and it can reduce or oxidize heavy metal ions and then aggregate them and remove them from water. There are actually already commercial products of graphene that we can use in water filters. They're a little expensive, but the process for making large-scale graphene are becoming very effective, relatively cheap, so we can use them in water filtration. This is another example of nanomaterial used for filter or nanofiltration. This is also involving the cellulose. Remember our good friend cellulose, the nanocellulose? So we can actually make another assembly of cellulose. This is called hemicellulose. Basically, hemicellulose are many cellulose nanofibers that are organized like this or like this. And what it makes a very good membrane, okay, that can capture both pollutants or particles, anything that you don't want to have in your water. And when you pass water, polluted water, in this hemicellulose nanofiber, you can clean the water. Very nice. Now, let's talk about nanoparticles. In many cases, as we saw before, as we talked before lunch, nanoparticles, particularly metal nanoparticles, can oxidize molecules. So the oxidation that they can do can be both for organic pollutants, but you can actually also oxidized microorganisms like bacteria. So the effect of taking electrons and destroying molecules can be done also for entire microorganisms like bacteria. And then what you get nanoparticles, particularly metal nanoparticles, that are antimicrobial. This is very important because in some cases we have contamination of bacteria in drinking water. So you can take the water, you can mix it or pass it through nanoparticles, through metal nanoparticles. These metal nanoparticles will oxidize the bacteria and kill them. Very good. Let's talk about magnetic nanoparticles. How do you think we can make magnetic nanoparticles? What elements we can use for magnetic nanoparticles? Iron. manganese, all the metals that form magnets. Indeed, we can take iron and make small nanoparticles of iron, and then these nanoparticles will be magnetic. Now, why it is good to have magnetic nanoparticles? If we have magnetic nanoparticles, we can control them externally with a magnet. Okay, just like we have here. If these nanoparticles will be magnetic, we'll have a magnet, in this case, outside this container, and all the nanoparticles will come towards the magnet. Very simple. What we see here is actually a very simple way to clean water. Because what do? Let's say we have polluted water. Water with lots of bad molecules, bad particles, whatever. Then we take and put magnetic nanoparticles inside this water. Okay? We put it in the water, we mix it, we shake it. The magnetic nanoparticles will absorb various contaminants through the fact that there is surface area or oxidation, and all the pollutants will absorb on the surface of the nanoparticles. Now the question is, okay, so we have water, we have pollutants absorbed on the magnetic nanoparticles. Now the question you can ask is, okay, but now we have to clean not only the pollutants but also the magnetic nanoparticles. How will you do that? This is why we have the magnet. We take the magnet, put it from outside, all the magnetic nanoparticles with the pollutants absorbed on their surface, they will attract to the magnet, and then we take all the water, we pour it out, and this water will be pure. So the fact that we are dealing with magnetic nanoparticles give us a way to purify the water from the magnetic nanoparticles and the contaminants. This is actually a common technique that people use in industrial settings. So this is, for example, magnetic nanoparticles with contaminants. We can pass the water through this container. Then we're going to put a magnetic rod. Just put a rod inside the water. All the magnetic nanoparticles and the contaminants will stick to the rod. And what we have here, just purified water. Now, nanoparticles can be also very good photocatalysts. Remember, when we talked about photocatalysis, what we have is light generating electrons, light hitting the nanoparticles, giving energy for electrons to leave the nanoparticles and oxidize. What the light is doing is, when we have organic pollutants, the electrons that we generate with light, they will go and break the organic pollutants. This is big organic pollutants, big molecules, and after doing the catalysis, after breaking the bonds using light, we can get these simple building blocks that are not bad. Okay? So this is photocatalysis of organic pollutants. We can see it here, the concept. We have wastewater, dirty water, water with various organic pollutants. we put inside the water photocatalyst, nanoparticles that are able to generate electrons because we shine light on them. In some cases, actually, when you shine light, you can get oxygen from the air that gets reduced, and this oxygen is actually a very good catalyst. So the catalyst can be either the nanoparticles themselves or oxygen that becomes oxidative, becomes a catalyst, and following the activity of the oxygen or the nanoparticles, we break the organic pollutants and we get clean water. This is an example of photocatalysis, purification of water with light. Let's go back to the story of the carbon dots. Okay, and I'll tell you about two very interesting applications of carbon dots for water purification. So let's talk a little bit more deep on carbon dots. Before lunch in the morning, I gave you a very general introduction on carbon dots. Let's talk more specifically on their very interesting physical properties. So these are carbon dots. Basically, it's just like a ball made of these 6-member rings. Just like graphene. Remember graphene, the graphene sheet? So carbon dots are basically little particles, graphene particles. So actually, when people refer to carbon dots, sometimes they call them graphene quantum dots because the structure, the internal structure of the carbon dots is very similar like graphene. These 6-member rings, which actually means that also carbon dots will be conductive, electrically conductive. This is how the carbon dots look under electron microscope. You see very small dots, very small nanoparticles. And if we look closely on the carbon dots, you see these lines here. These are exactly the sheets of graphene. Okay? The same atomic structure, also the same organization of graphene sheets. Now, this is not why carbon dots are so interesting. Just remember, to make carbon dots, very easy. That's why we also like them. Because when you have an easy chemical system, an easy preparation, it's always better compared to complicated chemistry. So we can make carbon dots basically from any carbon source. Now, the first reason that people really like carbon dots are the colors. When people first discovered carbon dots about 30 or 40 years ago, what they found was that carbon dots have remarkable colors. You can get from purple all the way to red. Now, this is actually very strange. When you think of carbon, when you think of coal, for example, coal is also pure carbon. When you think of a pencil, you know pencil? You know what's we use for writing in a pencil? Graphite. Graphite is also pure carbon. Does graphite have a color? I don't know if they have a color. Graphite is black. Coal that you burn in power stations is also black. Suddenly we have carbon that has a color, just like a rainbow. This was really surprising The reason why carbon dots have color actually, and not only that they have color for the entire spectrum, is precisely because of the size. Because they are so small, you can have color. And this color is actually because of a very special property called fluorescence. Have you heard of fluorescence? These colors are fluorescent colors. So you get them if you shine light on the carbon dots. And the reason you get colors of carbon dots when you shine light is precisely because electrons can jump from low level to a high level. Now, these are not carbon dots, these are semiconductor quantum dots. Semiconductor quantum dots are the first nanoparticles that people discovered about 50 years ago. The color of semiconductor quantum dots is actually related to the size. If you have a big size, you excite the electron to a low level, it goes back, it gives you red color, and this one gives you blue color. But it gives you color. With carbon dots, actually, the color is related to the fact that you excite electron from the inside of the carbon dot to the surface of the carbon dot. When you excite the electron from the core to the surface, and because the carbon dots are so small, you can get visible color. And the reason you have different colors is because the surface of the carbon dots is not completely uniform. You have various chemical groups on the surface that gives you different colors. So this is very interesting. Does it have to do with water purification? No. This is just the reason why people became very interested about carbon dots. And actually, they can use the colors for various things. For example, sensing. You can use the color for sensing. You can use the color for labeling bacteria and cells, for example. But this is a different story. Our story is not color, but is related to the fact that when you shine light on the carbon dots, what you do, when you shine light, remember this slide, when you shine light, you excite the electron from low level to high level. Now, when you excite the electron from low level to high level, what does the electron want to do? Go back. In nature, always the desire is to go back to the initial stable sort of situation. So the electron, you excite it with light to a higher level, it wants to go back. The easiest way to go back for the electron is to jump right back. When it jumps right back, it gives us the color, these beautiful colors. And this is what happens in solution. But when you have solid, when you take carbon dots and they cannot move and they sit in solid matrix, either when you dry them or when you put them in some kind of solid gel matrix, then the electron that you excite cannot go back directly because of the solid phase. Instead, what happens is the electron will go back indirectly. It will go back from here to other levels that the electron finds in the environment. And this process generates heat. And And that's the key thing, as we're going to see in a minute, in these water purification applications. This is called a photothermal effect, light-induced heating. So remember, the key thing, the important thing is carbon dots in a solid phase, they don't have color. But when we shine light, they generate heat. This is called in the physics language, multi-phonon radiation. This is a thermal effect. Okay, so we have a photothermal effect. Let's see how it helps us. And what I'll show you are two interesting applications of the photothermal effect of carbon dots for water purification. Now, why do we need water purification? Because water is, or finding clean water is not so easy these days. Either we live in a desert, or that there's not enough water, or if we live in places where the water is polluted. In both cases, we don't have enough drinking water. We need drinking water. Now, there are a lot of water purification technologies these days. People use what's called reverse osmosis. There are membranes here that when you pass the water, you clean the water, very nice. There are ion exchange, something that you use in your labs. When you want to clean water, you pass the water in big column. That's how you clean the water. So if we have these technologies, what's the problem? Why do we need something new? The problem with these technologies is that, for one thing, they can be very expensive. For example, this technique, reverse osmosis, needs a lot of energy, a lot of electricity, which is expensive. This technique is usually not so efficient. So you have these big columns, you need to replace them every few months, for example, and there's other problems. So there's always a search for new technologies. Desalination, that's what we want. Let me describe the system that we made in our lab for water purification or water desalination. Now what we do, we took carbon dots, we mix them with two very cheap polymers. One is called chitosan. It's actually a very simple polymer that you can also find in plants. And this is CMC, methoxycellulose. It's also a very cheap and simple polymer. And when we mix them all together, we obtained a sponge. Okay? Just a sponge that you can look at it. Let's see if I have a picture. That's how it looks. Let's see, wait. No, I don't have a picture. But this sponge is very easy to make. The good thing about this sponge is that it can absorb water. So you can use it as a filter. Now what's the secret here? You have the sponge. We pass the water in the sponge. Water goes into the sponge, just like a membrane, just like a filter. And then we shine light. Now what the light will do, remember the photothermal effect. Light gonna get absorbed by the carbon dots. Now the carbon dots in this case are in solid environment. They are sitting in the sponge. Now remember, when the carbon dots are in solid environment, they release heat. We shine light, this becomes hot. It becomes hot, and more hot and more hot. What happens to the water when it gets hot? They evaporate, exactly. When you heat water, they evaporate, just like steam. The water gets evaporated. Now it's pure. There's no salts, there's no contaminants, there's no anything. You just need to collect the vapor, you just need to collect the steam, and you have clean water. Fantastic. Let's show you evidence that it's really happening. Okay? So we took carbon dots that can absorb light in a broad range. That's how it looks. Okay? This is actually the polymer that is porous. Looks like a sponge. So we see that this polymer is made of these big fibers. And if we look, it's very porous. There is lots of space for water to come in. Remember, we want a lot of water to come in because we want to clean, to purify a lot of water. Okay, so this is how it looks. This sponge without carbon dots, and this is with carbon dots. And the black color, the brown actually color, tells us that there are carbon dots there. We cannot see them in this picture because they're very, very, very small. The scale here is 50 micron, so they're sitting all over the place and they make the sponge black. Now look at this beautiful picture. What we see here is images of a photothermal camera. Do you know how a photothermal camera works? When something is hot, it gives us red color. Actually, photothermal camera allows us to take pictures in the dark, at night. Because, for example, when people or animals are outside in the dark, they emit heat, and you can see that with a camera. This is actually another application is in war. You have binoculars that are sensitive to heat, to IR. So what we see here, these are photothermal images of just this simple polymer without carbon dots. Remember, the more red color, the more hot. So here we shine light, and after 60 minutes, we see some red, and the temperature, 28 degrees. Do you think it's a high temperature? It's not such a high temperature. It will not evaporate the water. But when we look at these pictures here, this is this gel, this sponge with carbon dots. Remember that the carbon dots are doing the photothermal effect. So we irradiate, we shine light for 0, 15 minutes, 30 minutes, 60 minutes, and within 60 minutes, look at the temperature. The temperature now is almost 60 degrees. Much higher temperature than this gel without carbon dots. What we see here, these colors compared to these colors, is exactly the photothermal effect of the carbon dots. They absorb light, they make heat, they heat the gel, and the water will evaporate. Okay? These graphs show you how the temperature is going up when you have carbon dots inside the gel. Now, what can we do with this? What we see here are graphs of using this gel, this concept, for cleaning water. So we took now water with different contaminants, different ions, heavy metal ions, very bad for our health. But when we do this photothermal effect, this heating, we can clean the water almost from 100% of the contaminants. What we see here is removal of the heavy metal ions. We can also remove organic contaminants, organic pollutants. You can see here that after we make water with some organic pollutant, it's called SDS, but after passing in this gel, we get pure water. This is another example of an organic pollutant that has a color, a visible color. It's called R6G. You see very high absorbance. You get water which are actually red with this polymer, with this contaminant. But again, after we purify the water with our special gel with carbon dots, nothing. We cleaned the water completely. Very nice. Another story. Using the carbon dots for oil spill cleanup. Now what we see here, sometime, unfortunately, there is oil spill on the ocean or on lakes or on rivers. When you take oil and you spill it, it goes and covers the water, it kills the fish, it kills bird. It's really bad for us. You know how you clean oil spill now? It's very, very difficult. What you need to do, because oil is very sticky, what you do, you take people or a ship, okay, and you just dip some cloth or some sponge or something, and you just simply need to collect the oil. This is very hard work because oil is so viscous. So it just stays. It's hard to do physically. And there's no technologies that can clean oil from water very effectively. There's some people that put chemicals that break the oil, but this is not so efficient, and it can also contaminate the water with chemicals. Some people put bacteria, some technologies put bacteria, which is also very inefficient. So this is a problem. How to develop technologies for cleaning oil from water. And the problem is, or the core of the problem is that oil is very viscous. Very hard to work with this. Now let me describe to you a very interesting solution that we developed in my lab. Again, that takes carbon dots and specifically takes the photothermal effect of carbon dots to develop technologies for cleaning oil spill. The concept here is very similar to what I described to you before. We make a sponge. This sponge is actually even more simple and cheaper than the previous sponge that I showed you. This is called polyurethane. You can buy it in the store. It looks like this. This is a real picture. It's just a very, very soft and flexible sponge made of polymer. Very simple to handle, very cheap. You can buy square or meter cubes of this, very cheap. Why I emphasize this is cheap? Because when we want to use it as something practical for oil spill, for example, we need something that's going to be cheap and available in large quantities. So this is the polyurethane sponge. What we did, we add carbon dots with some polymer which acts as a cross-linker. And what we get is a new sponge with the carbon dots sitting inside the pores using the PDMS here as some kind of helper, some kind of cross-linker. The important thing is that now we have polyurethane with many carbon dots sitting inside. Now, you can see this is a real picture. That's how it looks. Carbon dots are kind of brown, dark brown, so we can see visibly that the carbon dots are inside. Now, the reason why we use polyurethane, there's two reasons actually. One reason that it's cheap. There's three reasons. One reason that it's cheap. Second reason is that it is very porous, meaning that you can have lots of liquid solution going inside. The third thing is that the surface of polyurethane is very hydrophobic. Why it's important for us to have a hydrophobic surface? Because we want oil. Oil is hydrophobic. So if we have hydrophobic surface, maybe the oil will go in, and then we take it and go home. But what's the problem? Oil is very viscous. So even if we take the polyurethane, the sponge here, it's very hydrophobic, and we dip it in the oil will not go in because it's so hard to move. It's so viscous. So what we need to do in order for the oil to come in, we need to make the oil less viscous. Do you know in the kitchen how you make oil less viscous? Certain oils, certain fat, how you make them less viscous? Heating. Try it at home. Next time you cook, think about it. When you have fat, when you want to clean the oven or something, you need to heat the surface or to heat the pan in order to make the oil less viscous. So our idea here is, let's do the same thing. Let's try to make the oil less viscous, and then it will go very efficiently into the sponge. How we can heat the oil? With the photothermal effect of the carbon dots. Okay? So what happened? We have the sponge that is very porous with all the carbon dots sitting inside. We put this in the oil. We have the ocean, we have the river. We dip it in the oil, and then we have the sun. We shine light. Okay? What the sun will do, the light will get absorbed by the carbon dots, and then we have the photothermal effect. The carbon dots are going to release heat to the environment. The oil will be less viscous, and it will go very easily into the sponge. Then we take the sponge, we do like this, and all the oil goes out. And we clean the oil. Okay? Let's see if it really works. Okay? This is an example of experiment we did in the lab. Okay? So this is the sponge with the carbon dots. What we see here, we put the sponge in the oil. We actually put little color so we see the oil more clearly. So we put the sponge inside the oil. Here in the top is without light. Without light, in 5 minutes, nothing happened. Why nothing happened? What? There's no light. The oil is viscous. It doesn't go up. What's happening here? Here this is with light. We put it in after 0 minutes, but after 5 minutes, the water is clean. because all the oil goes into the sponge. Okay? This is, again, another very beautiful example. This is water, and this is oil. Okay? We put the sponge in, we shine light, and then we can separate the water and the oil. We can actually do it many times, in many cycles. See, these 50 cycles, and what we can see here is this is the oil that was on the water, and this is the oil that we can take out with the sponge. You can see it's always almost identical. We can do it 50 times, which is very important for practical application. Because if we sell this sponge to somebody that wants to clean oil, it's not that he can use it only one time. He can use it 50 times, 100 times. It's very good for practical application. That's it. Questions? So you're going to buy it from me? These things for water desalination? No, I'm joking. I don't have a product yet. I'm not a salesperson. I'm a scientist. But maybe one day, these technologies that I showed you, maybe they will be used practically. Who knows? I'm sorry? Everything that I showed is commercially available. All the polyurethane, all the sponge, all the other materials that are commercially available. The entire thing with the carbon dots is not commercially available. This is still a lab demonstration. Nothing, I don't sell it yet. No, well, in my lab they're using the technology. But it's not that I sold it to somebody yet. But I hope to do one day.
Just like one of the slides you have shown, that CMC plus chitosan.
Yes.
So CMC is like a carboxy metal cellulose, CMC.
Yes.
So surface is hydrophobic, is supposed to. And the chitosan is hydrophilic.
Exactly.
So it's kind of composites they are doing.
It's a good point. Overall, the sponge is hydrophilic. It is hydrophilic because you want the water to go inside. So the relative concentration of CMC is not as high compared to chitosan and the carbon dots, and overall the sponge is hydrophilic.
So it just be mixed
Yes?
without any condition.
No, well, we add this ECH, which is a molecular cross-coupling. Yeah. I'm sorry? Yeah, I think so. I don't remember. You're a polymer person. Yeah, it is very easy, and you can make the sponge with that, using that.
So cross-linking is going to be physical or
Chemical.
chemical?
I think it is chemical, yes.
And we can reuse it?
This one, yeah, we definitely can reuse it. I think we tried maybe 100 times, 100 cycles. Yeah.
So that may lower the efficiency
Yes.
after using?
Yes. What happens is that over time, you get a lot of ions and pollutants absorbed inside, and it's not so easy to get them out. So over time, it's not going to be so useful. But that's okay, because it's very easy to make it.
Can't we rewash it again and again?
We tried it. We take the gel with the ions, we cleaned it with little water, and then we reused it. So overall, we could use it several times, but obviously over time it is less efficient.
Any question from student side?
So,
I have also one question.
sure. Not to heat, but to do the photothermal effect. Yes.
Whether have you studied whether the temperature or the intensity of the light also play important role in that?
Absolutely. When you have high intensity of light, you have more light absorbed, you have higher temperature obtained.
So in continuation to that, the interesting fact is that Bilaspur, where you are standing, is the ideal place to perform such experiments. Where in the summer temperature goes to 40.
You're right. Yeah.
So you may get similar work, but you may get even better results.
Yeah, and you can get dehydrated also because it's too hot.
What are the basic criteria for being a carbon bond? Like we have so many carbon electrons, like graphite, diamond. So what are the basic characteristics of these bonds? What is the size?
It's much easier to make carbon dots than graphite or diamond. Basically, you just take carbon source and you heat it a little bit in 100 and 200 degrees, and that's how you make carbon dots. That's why it's so nice, because it's so easy to make them.
If we heat some compound, it finally becomes charcoal type of. So
You're right, but the difference between carbon dots and char is that here, which is actually very important, here you just heat it to 100 degrees, 200 degrees. It's not a high temperature. When you get char, you burn completely the carbon. You go to 500, 600 degrees. That's how you break everything and you get just carbon. But this is not carbon dots. Here, it's very gentle. You heat to a relatively low temperature, 100, 200 degrees, it's not such a high temperature, for a few hours, and then you get carbon dots. That's how you get them.
why this show this unique properties like photothermal? What is the reason why they are showing?
The origin of that is the nano size of the carbon dots. Because what happens is that you shine light, you excite the electron. And the electron wants to go back. And it goes back through going out in the material and releasing heat. Because when you shine light, you give energy. You give energy to the electron. The energy needs to go somewhere, and it's just heating the environment, heating the material.
And along with carbon dioxide, we make it also carbon nanotubes.
That's much more difficult to make. I didn't make carbon nanotubes. It's much more difficult.
Like practicing from biomass, you take carbon source. It is possible to make like graphene type material also.
Of course.
Low temperature like 250 around. It's also possible.
Look, I don't know how to make graphene or graphite sheets. I know it's much more difficult. I don't know the protocol how to make them, but it's not so easy compared to making carbon dots. It's not that you just take carbon and you heat it and you get graphene.
And even it is much more difficult to making it stable at the standing.
I think so, yes. But, at least what you have carbon everywhere. So you can make carbon nanotubes, you can make graphene, you can even make diamonds now. But much easier to make carbon dots.
It's possible if starting material contains some biomass like carbohydrates or cellulose, it may be paralyzed in temperature in 200 to 250. That may convert to light sheet structure like graphene.
Maybe, I don't know. I don't know. I don't work with graphene. I work with carbon dots.
What is the driving force there?
The driving force is that when you give 100 to 200 degrees, what you do, you break bonds. You break and you break and you break until you reach a stage where you have small nanoparticles. Very small. And then it's more difficult to break the bonds because they are very stable. So that's what you do. You just break and break until you get small nanoparticles. If you give higher temperature, 500 degrees, 600, then you can take everything, break it completely, and get char. But this is not what you get here because you are very gentle.
So that has to be precisely controlled, the temperature.
And not so much. That's why I told you you can make it at home. Because what we do is you put it, you heat it, then you need to purify it, which is, you do purification with dialysis or certain simple chemical means, and then you get carbon dots. Anything that you want. Any carbon source. That's the nice thing about it. That's why I show this slide that shows many different things with carbon. You can take the computer and make carbon dots. You can take milk and make carbon dots. You can take flowers and make carbon dots. Everything around us have carbon. Whatever has carbon will make carbon dots. What do you mean size limitation? Of what? No, the particles is nanoscale. That's the nice thing about carbon dots. They're very, very stable. We find that the protocol is actually very stable. In most cases, you get these small carbon dots. Of course, you can control. People work on controlling the size very precisely. When you optimize your protocols, you can make carbon dots with 5 nanometer or 2 nanometer or 10 nanometer. That's possible. But it's not that they make from nano to micro. It's actually very uniform. That's really the nice thing about it. Because you break everything and you get the smallest particles, which is the carbon nanoparticles. Okay. Sorry.
Question, then let's we can take a short break for.
Yeah, we can take a break, although we meet again and then I don't know what we're going to do.
Yeah. So one nice thing I will tell about Professor Lal, he is also associate editor of a very prestigious journal. So you can utilize this time, especially people from the research background, how to write papers.
Actually, good idea.
How to get a good journal, publish in good journal, what will be the theme.
Okay.
So you can mold the discussion even for your own benefit.
Good idea. Let's show a paper and I'll tell you what is my strategy for writing a good paper.
Yeah, so that will be a benefit of research scholars.
Good idea. Thank you.
Yes.
Yeah, but let's take a break first. Yes, yes.
We will be back here by 3. 15 minutes. on how to make a very good art in the science to get your paper published in such a prestigious journal. Yeah, please.
Okay, so the important thing is that there is no yes or no for what, how to publish great paper or not. It's, it's always a lot of gray area. Sometimes you do excellent job, but the editor does not get it, does not understand your point. Sometimes your paper is sent to somebody that is not in the field, so they don't know what's good or bad. But the advice I can give you is a general advice that maybe can help. The most important thing is you do what is interesting for you. You work what is interesting for you, and then you try your best to write a paper and submit it. So what I'm saying is that there's no really clear answer. Now, the first thing that you need to consider if you want to have a paper published in a very good journal is that you need to make sure or to do something that is really in hot fields or in interesting fields. For example, these days there is a lot of activity about alternative energy. So there's a lot of interest in hydrogen energy. There's a lot of activity in energy storage, in new batteries, and new technologies to make batteries. So if you work in these fields, this is something that can open doors for good journals. That's one thing. The other thing is, if you select something, some scientific project, some experiments to do, you need, or that's at least what I do and what I tell my students, try to find a problem that will make an impact. It will be something really new. It's more difficult. It might take more time, but then it will be important. Let's say you developed, or somebody developed, a sensor for ammonia. So there is an interesting technology for a sensor for ammonia. Now, if you take the same sensor and show that this can be used for ethanol, this probably will not go to such a good journal because the technology exists and you show application for ammonia or for ethanol or for something else, it's not a lot of impact. It's not new. But if you show that you get a new sensor that can be used for ammonia or ethanol or whatever, then this is more interesting. And if you have a sensor for PFAS, remember we talked before lunch on these bad chemicals, bad plastic in water? It's called PFAS. If you develop a sensor for PFAS, which is very important, then immediately people will be, and editors will be more interested because the problem is very important. So this is one suggestion. When you think of your project, of your scientific experiments, try to think of something that will be new. Everything is new, but try to think of something that will be very new. Not applying something to a different problem, but finding completely new solution. Again, this is very easy to say. It's more difficult to do, but if you really want to publish papers in really good journals, try to think about it. Don't do something incremental. Don't do something small. If you do some small advance, small step, small contribution, it will likely go to a less important journal. So try to think of problems that will be very interesting, very relevant to the reality of today, and very new. That's my first advice. Now let's say you found some problem that is very new. You got experiments that were excellent, and you're ready to write the paper and submit to a journal. Now, there's several things that you need to do in terms of a good paper for putting it together. The first important thing is what will be inside the paper. And this is actually also related to your project. This is something that I see in many cases as an editor. In many examples, people describe wonderful things. They have very good observation. They find good measurements. They find new batteries. They find new sensors. They have new hydrogels, new materials. It's great, but that's not enough. Something very important in good paper is mechanism. The mechanism in many cases can be very difficult because you need to think what techniques to apply. Some techniques, you might not have them in the university. When I have some problem like that, when I do in my own research, sometimes I go to a different university to do the experiments, or I send my students. Sometimes I even send my students to Europe to do experiments. Because having a good mechanism, what is the reactions, how the materials is strong, how the battery is working, how the sensor is such a good sensor. This is very important to make a good paper. Because just to present observations as good as they're going to be, you get fantastic properties. The material is very strong. The membrane is very porous. The sensor is extremely sensitive. This is very nice, but it's not enough. So you always need to think of the mechanism. How? Always try to answer the how it happens. This is really a crucial part of good chemistry papers, to find the mechanism. Again, it's not easy. Sometimes it's the most difficult part of the research, because you need to think what experiment I will do. How will I explain the experiment? Do I need to do 10 techniques? I'm exaggerating, but maybe you need to do this and that and different aspects. But this is something very important, and in many cases, this separates a good paper from a very good paper. Okay? Now, regarding these mechanisms, in many cases, not in many cases, in some cases, it's also good to have a computation aspect, some theory. Now, I don't know anything about computation. I don't know how to do theoretical chemistry, for example. But think about it that if you have people that you collaborate that can do calculations, this can help in many cases. It can give you a good figure in the paper. So editors, when they look at papers, they want to see how you address the problem from various angles. You need to explain the concept. You need to have good results. You need to have the mechanism. And sometimes you need to have nice figure of that you use computational strategies or computational techniques. So this is something that you should remember. It's very important to have the mechanism. Now, in terms of the structure of a paper, maybe I'll show you one structure of a paper, of my paper, and you'll see what I mean. I'll try to, 1 minute, I will try to find a paper that has a mechanism. Okay? See, let's try to find a paper with one of my Indian postdocs. I'll try to find it. I don't promise I'll find it because I have many papers, but I will try. Let's see. Okay, let's look at this paper, for example. This is a paper we published, I think, last year. It's a very nice paper. It's nothing that I talk here. You see, it was published in Nature Communication, which is a very good impact factor, I think 16 or something. This is something to do with biology. But let me show you what is going to be the best structure of the paper. Okay? Now, when you write an abstract, of course, I'm not going to teach you how to write an abstract. You write an abstract that will give you the main points of the paper. What I'd like to show you now is how you build the figures or the structure of the paper. Now, one thing, this is actually not such a good example, but one thing, in my opinion, that's very important in papers is to have many pictures, many schemes, because people like to see nice drawings. In our hearts, we're just like children. We like pictures. So usually in papers, the first figure I do, I make a drawing of the concept. What is the big plan? So for example here, you can see I make a little drawing that shows the concept of the paper. In this case, we have some antibiotics, we have some nanofibers, and these nanofibers, they break the antibiotic, they catalyze hydrolysis of the antibiotic. And this is very simple picture, very easy to see, and it explains your story. Okay, before you even show any results, just make a picture, make a drawing. Okay? So this is the first figure, and then we have some results. Okay, we did some results. That's the next step. Okay, you're showing all your beautiful results, very important results. I personally don't like complicated figures. I try to make it simple figures. And if you have a lot of data, try to separate into separate figures. Okay, so these are the results. And then remember, I told you about the mechanism. This figure is a mechanism figure. And remember, I told you about the calculations. This figure is a mechanism using calculations. But what I'm saying, this is very important that, again, very visually, we explain the mechanism. This is a molecule sitting like this. Then there is catalysis, and then they break. That's the other important part of a paper. If I can give you one advice, when you are ready to write a paper and everything is ready and you have great results, one figure, first figure, is the scheme of the concept. The last figure, a mechanism. Okay? So here you have a mechanism, and I think that's the last figure. Maybe not. Let's see. Yes? Oh, actually, we have more results. Yeah, but basically, that was the mechanism. Okay? Let's try to find another paper. Let's see here. I'm not sure if this one has what I meant. Okay, this is a paper. In here, again, you have a figure here. Actually, another important thing about papers in chemistry, if you have molecules, if you have new molecules, show the synthesis. Okay? So again, what we have here, this we have a new molecule. This is the concept. Okay, I explain what is the concept, first figure. Then we have the results. And actually in this case, and yes, and here we have the mechanism. Okay? What else can I tell you? Try to, if you're not so good about English text, okay, it's important to send it to somebody that can do editing, English editing. Because in many cases, editors and reviewers, if they see that the English is not so good, they think that the science is not so good. And it's not fair. Because sometimes you have great science and great papers, but maybe the sentences are not so good. So, you know, it's important to have really good text, good editing. So if you can, try to do some polishing, some editing, trying to have good text. That's also very important. Okay? What else? Don't make too long papers. Again, depending on your project, on the results, depending on the journal. I usually think that 6 figures is okay, 5 to 6 figures. You don't need a paper with 10 figures unless it's really something very special. Make a very focused story. Say in the beginning, in the drawing in the beginning, what's important, what's the discovery, what's the impact. Okay? That's actually also very important to mention it clearly in the abstract. Say in the abstract, we made for the first time this and that. We made a new material. that is best material for energy storage, something like that. Don't be too arrogant. Don't say something that we found medicine for cancer, okay? Because then people will say, okay, these people, they don't know what they're talking about. So don't be arrogant, but make a statement about why your work is so important and what's the impact. This study will make batteries which are very powerful. This work will find a material that is like the best water, not the best, an excellent water purification matrix. Okay, so it's very important that in your abstract and also in the conclusion, you clearly say why you should be published in a good journal. What's the impact of your work? I think that's my main advice. If you have questions, I'd be happy to answer. I hope I helped. I hope I gave you some ideas about good papers. Also for me, by the way, it happens that many journals reject my papers. It happens to everybody. I'm trying to have, I'm trying to give my students only projects that are new, really new, and high impact. But sometimes the editors of the journals, they don't think it's so interesting. They don't think it's such a high impact. So don't take it personally. Don't think that they don't like you. Sometimes it happens, but there's always a competition. There's many scientists in the world, and there's not so many good journals. There are many journals, but not so many very good journals. So it's hard. Okay? Yes. Look, it's all people. If somebody doesn't believe you, bad luck. If the reviewer does not believe you, but still the editor sends it to you for corrections, for revisions, then you can convince the reviewer that the experiments are real. And if he doesn't believe you, you can do more experiments, maybe a little different, and then you can try to convince the reviewer that your work, your experiments are correct. That's what you can do. Actually, it's a good point. Always when you write a paper, try to put as many references as possible for people working on the same field. Because people like to see, first of all, some people like to see their names in the reference list. And another thing is people want to see that you really was looking very carefully about what was done in the field, that you know what's going on, and you can convince why your work is so new and so interesting. So it's very important to do a very careful, very important, it's a good point that you asked that. Very important to do a careful literature search and to see what was done already. Actually, I had 1 or 2 times something that was very bad. One time a postdoc was working on a project. Always when I give people to start working on projects, I tell them, look, I don't know everything. You go, sit 1 week, 2 weeks, check the literature, check what was done and what was not done. So I told this guy, go and check. And he said, he was sitting and checked, and then he came to me and he said, nobody did this. It's new, what we were thinking of doing. And then he worked 1 year, and he got interesting results. And then suddenly, after a year, we had some discussion. I asked him, he showed me some results. I asked him, go and see if somebody did something similar. And then he brought to me a paper from 3 or 4 years ago that showed almost exactly what he was doing for 1 year. So basically this guy, he didn't do a good literature search in the beginning. So he wasted 1 year of his postdoc. So I also was kind of very, not angry, but I was kind of very disappointed because he wasted precious time. He did good things, but we're not going to publish it because somebody did this already. And as a postdoc, postdoctorate is very bad because your time is limited. So this is actually, following your question, that's another very important advice that I can give. Before you start working on something, check what was done. And if you see that people did something similar, go to your professor and tell him, look, it doesn't look to me so new. I want something different.
Doubting. Suppose you have produced 10 data or 20 data. If he finds even 1 or 2 data is something like not reliable, so he will start doubting on every data, even if the rest of the data is.
Things happen.
Yeah. In my Korean postdoc mentor, he always gives the same procedure, same reaction to some PG student to cross-examine the reproducibility of the. That also happens.
This is nice. Yes. Many reviewers don't care. They just read what you write.
Yes.
Good. So go and do the best science you can. Write excellent paper.
कितने लोग यहाँ PhD scholar हैं? Raise your hand. अच्छा. कितने लोग पेपर पब्लिश कर चुके हैं?
So the rest are not, what are you? You are undergraduate? Oh, masters? I see. So, but do you publish when you do masters? Do you need to publish a paper? Or even as a master's?
Project is there, but
You're not so sure.
So in the 4th sem we have dissertation we have.
Okay.
So but we want to be wished to and we are
Okay. Yeah, as a master you have short time, so sometimes you don't have.
It's not compulsory here.
Yeah,
Publishing is not compulsory. Doing project is compulsory.
yes.
But application sometimes
Okay.
it happens, sometimes.
Yes, of course, also in my institute. Okay, but after masters you can do PhD, yeah? So you can take this advice for your PhD.
In some cases, there is some retraction of the paper also.
Oh yeah.
Yeah.
In some,
Yeah.
yeah.
Because of, if something is really something breakthrough, they try to repeat the experiment.
Actually, what I have seen a few days back, that they have published on chemical review papers.
Yeah.
And there, 13, 14 reference, these papers have been retracted.
Wow.
And ultimately, the chemical review paper has been retracted.
That's,
Of
of course, yeah.
cross reference.
Yeah, that's bad.
Sir, I have one not doubt. Is I want to make more clear. Like some researchers, those who are working in theoretical chemistry or physical chemistry, they say means they have not good impression on impact factor. They say looping impact factor is something like worshipping false god. Even they sometimes say.
Yeah.
So what do you think? Is it really some marketing strategy or what, sir?
Look, it's a very difficult question. You know, in some fields, for example, in theoretical physics, for example, the impact factor of the journals are 3, very small, very low. And sometimes papers that get published in advanced materials, impact factor 30 or something, they're not so good. So, you know, people in general, they want some relationship. They want to have something to say, this is good and this is bad. And the impact factor give you something to measure. The ultimate thing is you need always to read your paper, a colleague from your field reads the paper and say it's good work or bad work. So there's no answer for this. You're right. It is in many aspects it is false god, as you say. It's a nice way to look at it. But it's very human that you always want to compare yourself or to have something, you know, this is a good thing, this is a bad thing.
And what would be better to provide more reference or less reference in a paper?
I think you don't want to have too much, too many. You don't have too little. I think it's always better to have more than less. If it's less, sometimes you miss some information. So that's my thinking.
Co-author, sir, in one critical physics paper in astronomy, I have seen 240 authors. That was the one
Wow.
collaborative. So
Yeah.
in our case, like experimental chemistry, means how much author looks like maximum.
Yeah, this is a good point. Actually, you know, in China, Chinese people, Chinese papers, you see 10 authors, 15 authors. Everybody is going into the paper. That's not good. I think you need to put 5 authors. I don't know. Put the people that really did something. Be generous. I'm always generous. This is not for a student, for you. But if somebody, a student or a colleague or somebody contributed something, put their name. But
Nowadays, they put a point that
Why you are wanting contribution?
more than 10 authors.
yeah, that's more than 10. I don't think is good. More than 7, I don't think is good. I think,
Contribution is there, then
yeah, if people did some experiment, that's my criteria. If somebody did spend time on experiments, then they can be on the paper.
Sir, to communicate with the students, so if we want to explore some possibility for the student, like international student, so whatever possibility for being a student in Israel,
Okay.
to have some internship program, so that would be opportunity.
Yeah. Okay, so the question was about opportunities in Israel for international students. So my university is very interested in international students in general. Now, because there's a war, and so now everything is low-key, hopefully in 1 year after the war, if there will be end of the war, then there will be a different things happening. Now, people in sciences, in chemistry, they're always looking for good postdocs, good students. So if you want, don't hesitate, if you want to go to Israel, of course, to do postdoctorate or even PhD sometime, you can just write. Write to the professors, find professors, write to them, and ask them if they can take you as a postdoc or as a PhD student. Now, this is something that I can really advise you. It's not only about Israel or about anywhere else, but when you write to somebody and want to show an interest in doing a postdoctorate or doing a PhD or something like that, don't send to 100 people some standard letter. You need to check people from the papers, from the web, from university sites. Find out if they're interesting for you. And when you write to them, tell them, I find your work on nanoparticles in water purification very interesting. I want to work on this. It's very important to show that you know what these people are doing. I, for example, get many applications from India, from other countries, from China, and when I see that whoever writes to me didn't really read my papers, didn't really, at least a little bit, didn't check my website, I feel that it's not serious. And then I don't answer. So this is an important advice for you if you want to go to Israel or elsewhere. Now, researchers in Israel are always looking for good postdocs and good students. So there's always opportunities. It might be more difficult sometime, but the opportunities exist. Yeah. So this is about individual thing. There are summer internship programs and also semester programs in Israel. Usually, it's university and universities together. So we sometimes we make an agreement with one university, MOU, one university with Ben Gurion University, with my university, to exchange students. Again, now it's very slow because of the war, but hopefully it will come back, and then maybe we can do something together, university to university.
There are some well-renowned philosophies like Deptt, DAD,
Okay. Yes, exactly.
Madam Curie fellowship.
Yeah.
Is
That's.
there some fellowship in Israel?
They don't have fellowships for short visits, There are very few fellowships for postdoctorate studies usually. There are information in the university site. There is a university site for international students. And there are some information about fellowships, etc. For faculty members, not with India. Again, it's usually university.
If they have some
Yeah,
collaboration.
if they have some relationship and they make contacts, then they can do a program for exchange, exactly, faculty exchange. But again, after my visit here, you know, maybe we'll do in the future some relationship.
That would be good.
Yeah. Yes.
So my question is, what is the benefit of doing PhD from countries apart from India? Like we see our teachers have done PhD from countries apart from India, and can we earn along with PhD? That is the best thing which we students can get. That we can earn along with PhD and we can continue with our studies.
So you're saying to do a PhD abroad, not in India? Yeah, I think it's a great thing to do PhD abroad. I myself did PhD in the United States, not in Israel. So if you can, do this.
So for that we have certain exams, or how can we?
No, there's no exams. Usually, again, you make contact with a faculty member, okay, by email. And you send your CV and you tell them, look, I'm interested in your science, in your research. And then the best thing, if you did something in your master's that shows that you're good, if you, in this particular field, if you publish papers, if you went to conferences, something like that. And then it's a question of luck. You know, if you have a contact with a person that has money and he agrees or she agrees to take you for a PhD. Of course, you always need to they always need to check whether you're eligible. For example, you're a chemistry master student, chemistry and biology. Okay. Sometimes they check what courses did you make in your bachelor degree, in your master's. Sometimes they tell you to have few more courses in your PhD. But usually if you have some chemistry background, they can take you easily for chemistry PhD. In my experience, for example, over the years, I had very good Indian PhD students. I had like 3 PhD students from India, and they were excellent. One was not good. It wasn't that he was not good, but he wasn't that interested, and he left after several months. But 3, including now I have 1 PhD student from India, and she's fantastic. So it depends on the people. If you find good connections, then it can be very nice.
Along with PhD, actually we have net examination with CSI and NET.
No, you don't need to do exams.
Yes, sir, but along with PhD, can we earn like any substitute for earning for students?
Money, you mean salary? Of course. PhD students, at least in Israel, they get fellowship from the advisor. It's like you need to live from something. So, I don't know, it's not a, I think it's good salary in Indian standards, and people in PhD students in Israel, they live from this salary. There's definitely a fellowship. For even for master students, there is a fellowship in Israel. Well, it depends. Because you don't speak the language, then don't let you. Israeli PhD students can get more money as a teaching assistant because they speak the language. You don't speak, you don't speak Hebrew, obviously, so they cannot do teaching assistants. There were some few cases that PhD students from abroad were lab assistants, and then they can make more money. They can get higher salaries. But usually foreign PhD students, they just get fellowship for research from their supervisor.
And even they can work in some restaurant or something somewhere else.
Well, usually, no, because you want the PhD students to invest all the time in research. But this is something that can, usually the fellowship that people get, PhD students get, foreign PhD students get, is okay for living. It's enough. And of course, there's always some prizes and awards for good students that can increase the salary. Yeah.
So while we are getting some new highlighting results and want to publish in good journal like JACS and top journals, so they are most commonly the editor or from reviewer we gave the question is not novel. So there is a novelty issue. So how to address that question?
That's the thing. It's very important. That's what I said about in the abstract. Very important to clearly emphasize the novelty of your work. Of course, the editors of such journals, they want novelty. But that's your job to emphasize it and to explain why it is novel.
Suppose sometimes based on the reference also it is said.
Look, that's the best you can do. Maybe the editor will like it or maybe not. The best you can do is to do a good job and to explain very clearly why it is novel. And that's what you need to write in the abstract, usually.
GGV is a good occasion that we are interacting with you.
Sure.
Just for a curiosity, so like as you are dean of research,
Yeah.
so this yourself can have our organization can have MOU with you, or you have to get permission from your own authority? Or
Yeah, usually I don't do MOU with foreign institutions. There's another vice president for global engagement. That's her job, but I'm involved in that. It's very straightforward.
How about this Hebrew is this learning Hebrew means
Oh, learning Hebrew is very difficult. Yeah, it's a difficult language.
after Chinese the Hebrew is
Well, everybody. I think Hindi is very difficult, but it's because I wasn't born here.
like regarding MOU, we have almost similar situation because he is also former dean and head, but he is still also not capable to do
Yeah.
the same thing. Again, he has to get the permission.
Okay.
actually
Well,
everywhere administration is involved.
But US, I think professor in the US, professors they have direct
oh, really? Okay. Didn't know that.
International
international offices involved. But generally, they recommend the international office.
Anything else?
Or any questions? In US, there are some chair professors, big professors, who's big professor,
Yeah.
chair.
Who? Myself?
In New York,
Yeah.
I'm
Okay.
But I know little bit about it.
Mm-hmm. Okay. Yeah.
So in Germany, like as a professor himself,
Yeah.
they can have the qualification.
Yes, I guess so.
On the German faculty.
Exactly. That's in Germany. In Germany, they're like kings.
They're actually, I would say, mainly resourceful, powerful.
Yes. Yes. Okay.
So it was extremely a good discussion with all of you. So you should have some ideas, some curiosity. Definitely you should have some curiosity regarding, and it's a really good team, okay, about your future, what you want to do after master program, after PhD, right? Those are your master program. So what is there, like a, the destiny, what is their future going to be, right? Because in Indian system we have, after master you have to clear NET or GATE exam, just to be enrolled in the PhD program or directly qualify the DRT exam. But except these things, or beside these things, we have also other opportunities. That you should be curious, right? Like international level, what's going on, right? Like regarding your question, so if you want to go to abroad, doing PhD, so then the common exam GRE, TOEFL, most of the students they have to qualify, like for US, for Canada, Australia. So keep in mind those things, right? Okay, I think that was a really fruitful discussion with you. Great discussion, and we thanks Professor Raj for this. And you have to be here all the time, tomorrow and after tomorrow, and just keep, writing some important tips, some important notes in your notebook, and don't forget these things easily, right? I think material probably.
No, online.
Okay, there is a link online, the YouTube link, and that will remain active, right? So you can watch at home also. If you have missed something, some portion, I think material will be provided to the students, right? Yeah. With that, okay, we thank you, sir, for everything.
Sure. Mm-hmm.
Yeah. He is.
Okay. Yeah. Oh.